So I'll try and go through this a bit quickly because I'm standing between you and the pub. So my talk is called Metahaplome. I'm Sam Nichols. I'm a second-year PhD student. I'm having a bit of an identity crisis after Ben's talk now as to how to introduce myself as a computer scientist or a computational biologist. But I'm working with Chris Creevy and Amanda, and you may remember me from some talks that I've given on the rumen microbiome. So for those of you who aren't familiar, this is a cow, and this is its digestive system. And as we've heard today, the rumen is the first and largest chamber in its stomach, and this is the one that's of interest to us. But what is so interesting about a cow's stomach? So this is an image from a microscope. These two big things that you can see are protozoa that mill around feeding on other bacteria and on food matter. And what you can't see in the dark is quite a considerable number of bacteria. And it's estimated for every milliliter in this fluid that there's between 50 and 100 billion bacteria. And what's so interesting about them? Well, they're constantly pumping out enzymes into their environment, whether it's for dominating resources that are available or trying to kill each other. And these are useful to us too. I mean, Ben's already outlined that we're all going to die because we're going to run out of antimicrobials, and this is a source of them. So the problem is, how do we get to them? They're not very easily culturable. They're not isolatable. So we turn to metagenomics. And people sort of wonder, why is metagenomics so hard? It's just that the sequence needed to make these enzymes is just found on some region of some DNA in some organism. But it's a gene, and it's on a genome, so I mean, you have to find it to start with. But that gene is not the only variant of that gene in that species. It's not the only species that has that gene, and those species also have their own variation. And that variation isn't uniformly distributed, and neither are the species. And this doesn't even touch on the species that don't have the gene that you're actually interested in anyway. And this doesn't touch on sequencing error and bias and sample bias. So this is all a bit of a mess, and this is the sort of thing that keeps me up at night wondering why I'm still doing research. So the story doesn't get any better, because with single organism sequencing, when you get your reads, you can turn them into sequences or contigs. And that's fine because they all line up against each other, and you can use the fact that some of them overlap to create genomes. And that's great because they all come, hopefully, from the same species or the same organism. We don't have that in metagenomics. So when you do your assembly, the contigs that you get out aren't actually anything meaningful at all. So these are effectively imaginary genomes. They don't actually exist in nature. They're just something that a computer thinks might be there. So when you find a gene on them, it doesn't actually exist. So if you look for genes that are similar, or whether you try and predict genes on a metagenome, they probably don't exist in nature. So this is where we're pushing for something called pseudo-references. So instead of referring to this as an assembly, it's a pseudo-reference, and you want to touch it for as little as possible. So you can look for a gene on your pseudo-reference, and once you've found it, you can take the reads that align to the same location of that region. And you know they align because that's what built them in the first place. And then you can do away with the contigs. So now you've got the reads, we don't have to pay attention to the assembly, and we've got an idea of what a gene at that region might look like. What can we do? Well, we can call for SNPs. That's what we like to do with reads. And then we look at the variation. Now, what we want to do is actually recover all of those different variants that we were talking about earlier, as many as we can, all of these different gene variants that exist in this metagenome. So we need to build these SNPs into some sort of structure that we can hold that variation in. And a big part that's quite important here is that the reads don't fully cover the length of the gene. And that's important because that means it's down to us to try and predict what these genes might look like without actually having a sort of a full example of what these real genes are. So how do we organize this variation? So I did what everyone would do, and I went to Belgium. I worked with a data mining team, and at first it was a bit of a struggle because I spoke English and they spoke mathematics, but in the end we settled on drawing pictures. And we came to these two different ideas. And the first was we could represent this variation in a graph. And you can think about that as, you know, a SNP is a street, and you're driving your car, and you just decide which path you want to drive down. And the other one is an association matrix, which is essentially just a list that says, read 1 contained this SNP at SNP 1, and it contained this one at SNP 2, and so on and so forth. But each have their pros and cons. And we wanted something that would kind of join both of the advantages of both of the data sets, both of the representations together. So something that came up while I was at Leuven is somebody suggested that we just try and go through the graph and find out how many paths there are, and we'll print them all out, because that'll be easy, right? So just before lunch, I set my laptop to do that, and about an hour later I came back. The room was very hot, the laptop was very upset, and back of the envelope calculation turned out that we definitely shouldn't have done that, because there were probably 2 times 10 to the 72 different paths in the data set that we had, which is a lot. So what did we do? We came up with this, and this is what we call the metahaplome. And the idea is that we encode the reads that align to some interesting part of the config that we've now discarded, and we can encode it into this graph. But we said that we don't really like graphs, we can't go through them, there's too many paths to enumerate all the possible options of the graph. So we need to be clever, so we decided to probabilistically weight the edges. And I'm very sorry that I have to do this on Friday, but we're now going to talk about some stats. But it'll be fun. Oh, great. So consider a much simpler data set. So these are our 4 reads, and we're at some position in the metahaplome. And we'll say that it's yellow because the rest of them at that position are. So what we want to do is predict what might come next by looking at the actual evidence that's in the reads. And in this example, we see that 2 of the yellows are followed by a green and 1 is followed by a blue. So you might say, okay, 66% probability that it's followed by a green. And this is great, and it seems really simple. So why has no one done this problem before? It sounds really trivial. So the problem is we want to take into account the rest of the path that we have taken to build this gene. So now consider these reads, consider that this is red, and we now see that there's actually no evidence for the gene that we've built, even with just these 3 SNPs, and that actually it should probably be blue. And the problem here is that the further forward you go, the more you have to have these equations that cross-reference who should follow whom. And in an example data set, we have 37 SNPs, and the number of equations to work that out is quite infeasible for a computer when you want to calculate a significant amount of them. So it seems we're kind of hosed, but luckily mathematics saves the day once again, and we retrofitted a spam filter. And the key thing here is that there's an idea called naive Bayes, and that means that we can treat these variation points here as independent, so we don't have to worry about the relationship between red and green, or green and red, but we do need to worry about everything and its relationship between the black node that we're trying to predict. So instead of it looking like this, it looks a bit like this. And this is much easier to calculate on the fly. And this is important because we need to be able to do these on the fly, because we want to reweight the graph once we've finished generating one path. So does it work? Yes. So we've got I've written a program that trivially generates small numbers of haplotypes, and then the program that implements this retrofitted spam filter to try and work them out. And this graph is the thing that it throws out at the end to tell you that it's done a good job and we can all go home. And on the y-axis we have the identity or percentage identity of recovery of a particular gene that we know is in the data set, and we know it's in the data set because we generated it. And on the bottom is the iteration number. And this graph is exactly what you want to see. Stuff hits 100 and then falls down again, and then something else hits 100 and falls down again. And this is fine, but sometimes it doesn't work so well. This is a data set that I'm trying to diagnose what could possibly be wrong. It seems to enter a very strange periodic state where it repeats what it's doing over and over again. Don't tell my supervisors. So why are we doing this? Well, metagenomic assemblies are problematic for reconstruction, so we've said that because they don't actually relate to anything that exists in nature. But it can be a pseudo-reference for probing sites of interest. So you might want to look for matches on it and then pull the reads that built areas around those matches in, because those actually contain the data that was observed in nature. Sequencing error and all that notwithstanding. So these paths that we create through the graph, they're built from real evidence. So these actually represent potentially real genes. So just going back to this example, you see that one line does actually start on the first iteration at 100%. So even though the rest of the graph is doing who knows what, we do recover one gene, and that's 37 SNPs long, and it actually corrects the reference, the pseudo-reference, in 12 places. So we can actually pull out that gene as it exists in nature from a data set that tells us that's not what it looks like. So that's pretty cool. So what is the metahaplome? Well, until we took it to a conference, it didn't exist. So if nothing else, we've created a word of our own. So to summarize, there's exciting enzymes hiding inside metagenomes. Metagenomic sequencing is definitely quite hard. We've shown it's possible to actually take evidence that you've seen on reads and put it into an interesting structure called the metahaplome, and that graph is too large, so you have to do something clever with it. So we've chosen to go down naive Bayes, but there's potentially other options. So what's next? Always tweaking, always optimizing. The current focus is on reweighting the graph because it seems that one of the largest aspects of actually successfully pulling out genes is not so much how you build the graph in the first place, but once you've got your first gene, how you reweight the graph so that when you go through it again, you don't get the same thing back out. So we'll be experimenting with different methods. We've also been testing this data set, this solution on a data set of limpet gastrointestinal reads, and as well as testing it in silico with simulated metagenomes. But ideally, what we'd like to do is, when we're a little more confident in our predictions, we'll use these as primers and go back to the original DNA sequences that our data set is built from and see whether anything can actually be extracted using those as primers. And also hopefully papers, because that's what we're all here for, right? So that's me. If there's any questions, I think we might have time. Otherwise, I'll see you in the pub.